The intestines of about 1/4 of the world's population have been invaded by these worms. They're parasites, and like all parasites, they cannot survive on their own. In order to live, they must invade the body of another creature, like man. From that host, they take food and shelter. In return, they give nothing. A parasite's objective isn't to kill its host. That would bring about its own destruction. The diseases parasites cause, therefore, aren't usually fatal. They're chronic, long-term, debilitating conditions. Man supports hundreds of different parasites. In the Western world, they're mostly harmless, but the parasites of the tropics can be truly terrible. These parasites cause an enormous body of tropical disease against which medical science has achieved very little. They affect hundreds of millions of people, and yet until recently they've been almost totally neglected. These scientists, however, are part of a counter-revolution against the parasite invaders. Gathered together by the Rockefeller Foundation for a conference, they're committed to doing something about these diseases, the so-called great neglected diseases of mankind.
Well, the great neglected diseases are called great because they're problems of usually hundreds of millions of people, hundreds of millions. And they're called neglected because they're neglected by the scientific establishment of the world, and neglected by the funding agency of the world, and neglected by the political establishments of the world.
No seafood is cooked.
But it's not going to be an easy task. The parasites which cause these diseases are formidable opponents, more complex by far than viruses and bacteria. They range all the way from tiny, microscopic, single-celled organisms, no bigger than blood cells, to worms. Many worms like to live in the human gut, where they feed on protein and blood. Ascaris worms can grow to 1 foot long. People can have up to 100 of them inside their intestines. Giant tapeworms are also found here. And probably worst of all is hookworm. 600 million people are infected with hookworm. Once inside the body, they cling to the wall of the intestine with sharp, cutting teeth. They live on human blood and cause a chronic, debilitating anemia. Not all parasites, however, are so fussy about where they live. In West Africa, there is a worm, the guinea worm, that will live almost anywhere in the body. The worms, which grow up to 3 feet long, often emerge as sores in the skin. The only thing to do is pull them out. These kinds of parasitic worms, guinea worm, ascaris, hookworm, thrive in conditions like these. The parasite infects the host at waterholes or from the soil, and then after growing in the host, it passes back through defecation to the environment, where it waits to begin the cycle all over again. But parasites like these, while distressing, are no match for man when he's well organized. Preventive measures, improved hygiene and good drugs can and have eliminated them in many parts of the world. It's the next class of parasites which has really defeated man's efforts at control. These parasites have expanded their life cycles to include another creature besides man, a carrier or vector. Usually a blood-sucking insect, the vector carries the parasites from host to host as it bites them. No matter how hygienically you live, you can't stop insects biting you. Four vector-borne diseases between them infect hundreds of millions of people. One group are the filarial worms.
Well, the filarial worms are roundworms, all of them transmitted by blood-sucking insects. And there are two of major importance for human welfare. One is the lymphatic-dwelling parasite. It's got a terrible name, Bushraria bancrofti, which affects about 300 million people, mainly in the hot, humid, tropical areas of the world. The disease is manifested by elephantiasis, the big leg, fever, filarial fever, mainly people in Southeast Asia. The other filarial parasite, which in some ways is the most dreadful of all the human parasitic diseases, is onchocerciasis, or river blindness. And a lot of the parasites get into the eye and it causes blindness. There are some areas in West Africa where 30% of the total adult population are blind due to this parasite.
This is the filarial worm that causes river blindness. The insect vector which carries it is a simulium fly. When squashed, you can actually see the parasite moving in the abdomen of the insect. A few such parasites won't harm the fly, but when they're injected into man, the effect is devastating. Inside the human body, the worms collect together in hard nodules under the skin. Deep within the nodules, the females start to produce millions of offspring, microfilariae, which are released continuously into the tissues. As long as these baby worms live, they don't cause too much trouble. But when they start to die, after 2 years roaming the body, they cause a massive reaction.
This is after lots of infection.
Terrible itching in the skin, and in the eyes, soreness, and eventually blindness. In Africa, this disease only occurs in communities that are located near fast-flowing rivers, because that's where the simulium fly breeds. Hence the name, river blindness.
We get the same simulium in this country, in all our streams in Scotland and in Wales, but they don't bite man, so we don't have the disease. But we have the disease in cattle instead. Now that disease can only be controlled either by killing the vector in the running water with insecticides, and this is a formidable problem in a country like Africa. So there's a World Bank program there bombing all the rivers of West Africa with insecticides. Unfortunately, the vector has now developed resistance to the only insecticide which is really available for this particular job. And this is making it very difficult. Now you say, well, why not get rid of this disease by treating the people, because they're already badly afflicted? Unfortunately, the disease is caused by the dying parasites. In the natural course of the disease, the parasites die in the skin or in the eye, and they cause the disease. So if you treat the people and kill the parasites, you often make the disease worse. And we've been looking for a long time to try and find a drug which acts slowly, only kills the adult worms and not the immature worms that are mainly causing the disease. And this has proved very, very difficult. We haven't got one yet. that can be used for mass treatment.
The schistosome parasite causes bilharzia. About 200 million people have it. The worm gains access to man from the water. It literally burrows through the skin. Once inside, the parasite grows, moving freely in the veins of the liver or bladder and in the intestine. They mate and live for up to 40 years. The problem begins when the females start laying eggs, hundreds a day. These eggs squeeze through the wall of the bladder or intestine, causing chronic, long-term bleeding. The bilharzia parasite comes from the water, and so to survive, it must return there. When people excrete near water, the eggs find their way back. The parasites hatch out and swim off to find another host in which to complete their life cycle. In this case, it's not an insect, but a water snail. Inside the snail, they multiply, and finally they emerge back into the water as new worms. They are then ready to begin the cycle again by infecting new human hosts.
It is a terrible disease because it reduces the survival of man. A lot of people in the endemic areas die at a young age with chronic complications, which even include things like cancer of the bladder as a complication of the bladder bilharzia and terrible disease of the liver for both the schistosoma japonicum and the other one, schistosoma mansoni, that affect the liver. So it's a very, very chronic disease. It's been very difficult to control. And I say the snails are not easy to kill because they have an enormous biotic potential. If you leave 1 or 2 snails in the water, within a few months, they're back to the same numbers that they were before you added the molluscicide. And you have to add this molluscicide, the thing that kills the snails, forever. You have to keep doing it forever if you want to control the snails. To control the parasite in man, we now have 1 or 2 very, very good drugs, but you have to treat the whole population. And this is almost impossible in the areas where the disease occurs because they are in the most underdeveloped countries of the world.
Not all parasites, however, are worms. Some are very much smaller, like the one that causes sleeping sickness, the trypanosome. While it debilitates thousands of Africans, its main effect is on cattle. Africa could be one of the largest meat-producing regions in the world. The conditions are ideal, but for the trypanosome and its vector, the tsetse fly. The tsetse has a voracious appetite. It can drink its own weight in blood in seconds. Since the parasite depends on the insect, one obvious approach is to destroy the tsetse fly with insecticides. And throughout Africa, the only place where tsetse flies live, it's been tried. But the areas are simply too vast. Vector control programs have been very expensive and have had only marginal success. In some areas, they've failed completely.
It's very difficult to control them in the great savanna belts because the tsetse fly not only feeds on man, but feeds on game animals.
While these animals don't actually get sleeping sickness, They carry the parasite in their blood, forming a huge reservoir of trypanosomes.
That one is going to be impossible to get rid of unless you get rid of the game animals, which would be rather unpopular, I think.
Filariasis, bilharzia, and sleeping sickness affect millions. But malaria is the greatest parasitic disease of mankind because it not only debilitates, but can kill. About 1 million deaths a year. The parasite infects man from a blood-sucking mosquito, and once established in the host, invades the red blood cells and destroys them.
Malaria is the great killing disease of mankind in the tropics. It is the most formidable tropical disease. There are probably 1,000 million people living in areas where Anopheles mosquitoes are transmitting the most dangerous type of malaria. There are several different types of malaria, one which is an acute, malignant malaria, and there are others that are chronic, relapsing type malarias, all of them transmitted by Anopheles mosquitoes. A very, very wide range of Anopheles mosquitoes, hundreds of different species, each with its own different habitat. Very difficult to control in the environment, but most of them, fortunately, rest in houses at night after they've been biting the people. And so you can control malaria by using insecticides sprayed inside the houses. And there has been a great deal of success in the control of malaria in the subtropical regions. The Mediterranean area used to be full of malaria. There's no malaria there anymore, except a little bit in Turkey. Most of Pakistan, most of northern India used to be malarious. A lot of that is now clear of malaria, the kind of subtropical belt.
In the tropical areas of the world, however, there's been much less success. Part of the reason is that the mosquito vectors have become resistant to the traditional insecticide, DDT. But even worse, the parasite itself is fast becoming resistant to all the antimalarial drugs on the market. In some parts of the world, malaria is untreatable.
The biggest problem area is for resistance of the parasites to the drugs is Southeast Asia. Some people blame the Vietnam War, the fact that vast amount of antimalarial drugs was used by the American troops and used amongst the local population, so that the parasites developed resistance to the drugs that were used for prophylaxis. Now we can't use the drugs for prophylaxis, and we can't even use the drugs for the treatment of the people. But that resistance that started in Southeast Asia is now in South America, and it's already reached the East African coast and is spreading into Africa.
All in all, our attempts to control parasites have largely failed. But there's another way of dealing with infectious diseases, one that doesn't require insecticides or drugs: vaccination. For some diseases caused by viruses and bacteria, it's been a massive success story. You won't see this disease, smallpox, anywhere in the world now, thanks to vaccination. By 1979, the World Health Organization could announce its total elimination. Smallpox is caused by a virus. If we could vaccinate against diseases caused by parasites, then perhaps we could control them as well.
It needs the least cooperation of the population that you're trying to treat. If you can vaccinate them when they're babies and they're going to be then protected, that's great. If you're going to treat them with drugs, you've got to find them, you've got to get them to come to the clinic, in some cases to go to hospital in order to have these drugs.
And that's where these young scientists come in. Sponsored by the World Health Organization and the Rockefeller Foundation, they're planning new weapons, like vaccines, with which to fight the parasites.
Science Advisory Council situation, and this meeting is about
They were brought in for their knowledge of molecular biology, immunology, and genetic engineering.
We've gone to the best laboratories in the world, regardless of where they are in the developing world. And we found there are a lot of scientists, outstanding scientists, sitting around who really are closet altruists. If they were given the opportunity to work on the great neglected diseases where they saw an enormous opportunity not only to deal with problems of vast numbers of people, but an opportunity that's been lying fallow for very many years about which they could really do something, they jumped at the chance. And we got those people in to apply the most modern techniques of molecular biology, biochemistry, and immunology to dealing with these problems. And of course, what's happening now is because nothing's been done with them for 50 or 60 years at this level, remarkable things are happening. Also, because of something we couldn't have predicted, and that is that the state of the techniques for biological research have suddenly taken off in a remarkable fashion in terms of genetic engineering, hybridoma technology, monoclonals, and so on.
A new breed of scientist has invaded the cloistered world of parasitology. Mostly they're not parasitologists. Before this new initiative, few had ever seen a parasite. Fewer still had ever been to the tropics. The old tools, vector control and chemotherapy, hadn't succeeded. They were out to make new ones. Above all, vaccines. A vaccination is a way of giving the body advance warning of a disease. Large amounts of the actual organism are pumped into people, but in a safe form, either weakened or dead. The immune system is now prepared, and the next time the invading organism tries its luck, the immune system swings into action and the invader is surrounded and killed. In principle, this should work for any organism, bacteria, viruses, and parasites.
Now, our problem in parasitology is that nobody has yet been able to develop, or until the special program started, nobody had been able to develop an effective vaccine which was using a dead product. And people don't like the idea that we're going to put live, living parasites in them to protect them against infection. We've all the time been searching not for an attenuated live vaccine, but for a dead vaccine.
Vaccines made from whole dead parasites had been tried, but didn't work very well. And the reason was simple. It's only a very small part of the parasite which is important in stimulating an effective immune response. The rest doesn't matter. A scattering of molecules on the parasite's surface antigen molecules, are the only parts which trigger the body's defense.
The parasite is a mosaic of antigens, many hundreds of antigens, which are inducing immune responses, most of which are totally useless, if not harmful. Some of which, we know, are useful and spectacularly successful in eliminating the parasite from certain individuals at least. Now, if we can identify those molecules, that small subset of molecules, that should form the basis of the molecular vaccine.
Any parasite invading the body will be killed off only if the immune system can recognize these few surface antigens on the parasite's surface. They are the Achilles heel of the parasite, the chink in its complex armor. These molecules are too small to be identified even with the most powerful electron microscope. But the body can see them, and if it does so in time, it can send large antibody molecules to stick to them. The parasites get surrounded with gluey molecules, and then the body's killer cells come in and finish them off. A vaccine made up solely of a lot of these surface antigen molecules, which cause a strong immune response, should be both effective and safe. It's a whole new idea in vaccines, and to make it happen requires the techniques of genetic engineering. First, you must pull out the genes of the parasite. The genes all code for different parts of the parasite, including the single gene which makes the key surface antigen molecule. But they must all be put into bacteria for cloning, because single genes are too small to tell apart. Bacteria are chosen because they can be tricked into accepting parasite genes as their own. Each of the hundreds of different gene fragments are taken up by the bacteria. And the bacteria are then grown up or cloned, copying not only their own genes, but the extra parasite gene as well. Bacteria divide very rapidly. The genetic engineers grow up hundreds of bacteria colonies like this one, each one expressing a different parasite gene. To find which one of these colonies is making the right surface antigen, they are tested to see which one will react with the antibodies in the blood sera of people who are resistant to infection. This colony will then form the basis of the molecular vaccine. If it sounds complicated, it actually looks quite mundane. The genes are extracted from the parasite by force. All the different genes are mixed up in a special solution of bacteria in a test tube. The bacteria are grown in culture, and the hundreds of different colonies are stored in a gene library, in the hope that one will come up trumps and react positively with the blood sera.
If by genetic engineering you can insert the gene into a bacterium which will multiply and grow very quickly, you theoretically at least finish up with very large amounts of this antigen, completely harmless and very effective. Well, that's the story.
The problem is that parasites also know some clever immunological tricks, so anti-parasite vaccines may not be so straightforward. Take the Bilharzia parasite. After it penetrates the body, it can live in the veins for up to 40 years without being rejected. How does it manage to conceal itself from the body's immune response? One widely accepted view is that it's able to disguise itself. Somehow it manages to coat its surface with protein from the host. Within 3 days, it's covered up its surface antigens from the body's immune system. It's disappeared. The body can't see it, so it can't reject it. Any vaccine against Bilharzia, therefore, would have to work before this camouflage was complete. The African trypanosome has an even cleverer strategy. Amazing as it may seem, it's able to vary its surface antigens to evade capture. As the body recognizes the surface antigen and sends antibody molecules to cover them, so the parasite population is able to change its antigens.
And so the immune response is left floundering, say, 1 week behind. By the time the immune response has sort of gathered its resources to attack a wave of parasites in the circulation, the parasite has evolved and it has changed itself subtly, so that the immune response is left floundering, having dealt with the one wave, and now having to mobilize its resources again to catch up with the next wave. So that is what we would call antigenic variation.
With such tricks, what chance is there of a vaccine?
Trypanosomes with their ever-shifting antigens, I don't think there would ever be a conventional vaccine. You might invent a drug which would stop them changing, and then the immune response could easily kill them. Schistosomes, intermediate. This disguise is a problem because you've only got a limited time, perhaps, in which to kill the parasite. But again, you might invent a drug which would work together with the immune response and perhaps catch them that way.
What of the other parasites? They also have ingenious mechanisms to evade the immune response. The Leishmania parasite actually hides inside the very white blood cells that set out to destroy them, the macrophages. Coated with a protective chemical, it enters a hiding place where the rest of the body's immune system would never think of looking. Other parasites live in parts of the body which are out of reach of the immune system, like the gut.
The parasites in the intestine are sort of protected against much of the immune response. They're in a sort of privileged environment, immunologically privileged site, if you like. Other parasites produce products which are anti-inflammatory. They sort of suppress the expression of immune responses, or they cut antibodies in half. One very effective means is to produce molecules, enzymes, which cut the main effective arm of the immune response in half. So any antibody which is crazy enough to bind to the parasite finds itself cut in half or split up into little pieces by the enzyme liberated by that parasite. So obviously a very effective way of neutralizing one of the major arms of the host protective immune response.
How are parasites able to do these remarkable things?
Well, I think they've learnt to do it. They've learnt to do it in the Hart School of Evolution, and they've become very good immunologists. They have developed ways of evading the immune response, many different ways and some extremely elegant ways. Those who have succeeded in evading the host immunity are the parasites we see. Those who haven't have been long extinct. It's a very, very hard school. I'm quite certain if immunologists had to work in a school as hard as this, there wouldn't be so many of them as they presently are.
Attempts to liberate people from the filaria parasites, which cause river blindness, must take account of another problem. Inside the body, the growing parasites go through different stages, displaying different antigens. The adult worms are slightly different antigenically from the baby worms they produce, the ones which cause the disease when they die. Telling them apart is immunologically very difficult, but it's crucial. Develop a vaccine against the wrong stage of this parasite and you might cause disease, not prevent it. A vaccine which caused the sudden death of thousands of baby worms in the skin and eyes could be fatal. Of all the parasitic diseases, scientists agree that the best chance for a vaccine is in the most deadly, malaria.
I think, first of all, children do recover from malaria, so there is a good ongoing immune response. Secondly, I think malaria is probably the worst parasite at evading immune responses. It doesn't quite have the same clever tricks as schistosomes and trypanosomes. So I think it's a better target. And then I think, thirdly, the molecular biology approach, which we were talking about, is much further on in the field than in others.
The prospect has attracted scientists from all over the world, and they're not all going about it in the same way. There are 3 distinct stages to the life cycle in man of the malaria parasite. The infection stage, when it gets injected into the bloodstream, the so-called sporozoite. The stage in which it destroys the blood cells and causes disease, the merozoite. And the sexually mature stage of the parasite, which is picked up and transmitted by the mosquito, the gamete.
Well, the situation here is very interesting. And a good illustration of the complexity, the life cycle complexity of a parasite. Malaria goes in as the sporozoite, from the mosquito goes to the liver, escapes from the liver into the blood, multiplies, and then is taken back by the mosquito to perpetuate the cycle. And malaria vaccinologists, if you like, are focusing on these 3 stages of the sporozoite, which is being introduced, so anti-infection immunity or vaccination, the stage in the blood, which gives you the disease symptoms of malaria, anti-disease stage vaccine, and then the transmission, the stage which is transmitted, the gametes, through the mosquito, anti-transmission immunity.
Most scientists are working on the disease-causing merozoite stage. After early successes, however, they've run into trouble. The experimental vaccines, which were being tested in monkeys, only seem to work if mixed with a toxic additive, unacceptable in man.
One of the other big problems with the Merozoite vaccine is that it's only species specific. So it would only be effective, well, all the malaria vaccines would only be effective against one particular species. And man is infected with 4 different species of malaria. The other problem is, rather like trypanosomiasis, it's recently been discovered that the malaria parasites also undergo antigenic variation, so that within a species there are many strains and many variations, and it may be that one type of vaccine developed from one species or one strain would not protect against others. So if we use an African parasite to develop our vaccine, it may not have any effect against the parasite in Southeast Asia.
Much more promising in the short run is the work on the first infective sporozoite stage. Examination of the parasite in this form shows it to be covered with an identical surface antigen, which provokes a very strong immune response. Sporozoite antigens from different strains of malaria are very similar and don't seem to undergo antigenic change. Last year, a team at the New York University Medical Center successfully made an antisporozoite vaccine. They did it for a form of malaria which affects monkeys, and it worked. This year they're repeating it for malaria falciparum, a form of human malaria which kills 1 million people a year. They already know which surface antigen is important. After extracting the sporozoite parasites from the glands of infected mosquitoes, they will, in the normal way, clone the parasite genes in bacteria and grow up a colony that is expressing this surface antigen. If all goes well, they'll succeed by the end of 1984. The others are watching closely. If they succeed with malaria, then other parasite vaccines too may in time be possible.
A vaccine that could be used for people visiting endemic areas will probably be the first one to be produced. It could then be used in areas where malaria control campaigns are taking place to protect the people after there has been malaria control, to protect them from reinfection. I think that will come within the next 5 to 10 years.
There is a problem, however. Once the mosquito injects the sporozoite, the immune system only has about 30 minutes to catch it. After that, it's safe inside the liver. So however good the vaccine, if even one sporozoite gets through, the host will get malaria and pass it on. But there's a more serious problem.
There are enormous snags in the whole vaccine development. Somebody has to be encouraged to try and develop it commercially. And there's very little, I think, chance of one of the big industrial companies taking on the development of a malaria vaccine. There's no profit to be made from tropical diseases. And this is the reality. You can make more money from the production of a new tranquilizer or something to cure a headache than you could make from all of the tropical diseases. Because the people in the developing countries can't afford to pay for the drugs, and they won't be able to afford to pay for the vaccines. So if they're going to be used, they will have to be used with somebody else buying them, and that means that the world community, through the World Health Organization, through the World Bank for Development, will have to pay for the vaccines if they're going to be used.
But there may be another way of making it profitable.
That there's an enormous movement of tourists throughout the developing world. Germans are going to Kenya, and Swedes are going to West Africa, and Americans are going to the West Indies and so on, and to Mexico. And these people are facing problems like malaria. It's approximately 5 million American tourists per year go to the developing countries where malaria is existent, and these people need to be protected. And as I pointed out, the drugs are inadequate.
Nevertheless, last year, attempts to get the US biotechnology firm Genentech involved in developing a malaria vaccine ran into all kinds of difficulties. Genentech couldn't see much chance of a profit for the time being at least. Ironically, there are already vaccines against parasites which are very profitable. This is one. Unfortunately, it's not for humans, but for animals. In sharp contrast to the human situation, veterinary parasitology is big business. Millions of pounds are spent by farmers every year to keep their livestock healthy.
Much more money is spent on developing drugs and vaccines to protect livestock than to protect man in the tropics. But there is a spin-off. Quite a number of our more exciting new drugs that we've got for treating people have come from veterinary parasitology research institutes. The development of the vaccine started in veterinary medicine, and we now have vaccines that are used in this country to prevent coughing disease in calves.
Husk, or lungworm, is a devastating cattle disease common to cattle all over the world. Parasitic worms infect the animals and make their way to the airways of the lungs. They're not found in the upper part, but lower down they're everywhere, blocking the passages, making breathing very difficult. Every other means of eliminating this parasite was tried, but only the vaccine succeeded. The cruel fact is that in commercial terms these cattle are profitable and the human beings of the third world are not.
One course of Dictol for every new calf leads to lifelong protection from the expensive after effects of husk, poor growth, poor finish, delayed bulling, and all the associated extra feed, labour and housing. Husk is expensive, but Dictol isn't, and it's very effective.
The development of anti-parasite vaccines for humans, therefore, will have to be paid for by the world community, just as for smallpox.
But that's only the beginning of the problem. If we have the vaccine, we manufacture the vaccine, we can supply it to the people in the field. Doesn't mean to say that it's going to work. We already have probably one of the best vaccines known to man, the polio vaccine. If you go to Africa, you'll see plenty of cases of polio. We have a measles vaccine. Measles is one of the great killing diseases of Africa. The malaria vaccine won't be perfect. It may need to be given every 6 months or every 12 months. It won't be long-acting like the vaccines against yellow fever and smallpox. If that's the case, the logistics of the delivery will be difficult. So it will be a very big expense on the local health service in terms of manpower and transport. So we mustn't kid ourselves that if we have a vaccine that we're going to get rid of malaria with by vaccination, we're going to still have to control the mosquitoes. We're still going to have to treat people with malaria using drugs.
It no longer looks as if the wonders of molecular biology alone are going to win the war against the parasites. The parasites are too well established, their defense mechanisms too sophisticated, and their hosts are too poor. But vaccines, if and when they come, may shift the odds a bit in our favor and reduce the amount of human suffering. Parasites are much more subtle creatures than was once thought, but they're not infallible. Their complicated life cycles mean they can be attacked at many points. Each individual attack may fail, but a concerted effort from all sides with all the weapons, drugs, insecticides and vaccines may, in the long run, have a chance.
Well, I happen to be a great optimist in that I believe that mankind, men and women, are smarter than mosquitoes and worms.
Or are they here to stay?
I would think so. People talk about the eradication of smallpox. Well, smallpox was a very, very stupid organism. It stayed exactly the same antigenically ever since Jenner's day. It didn't have any reservoir hosts. It was purely in man, and it either killed the man or the man killed it. It was an acute infection, nothing chronic. I think parasites would be very much more difficult, but of course we've got to try, and I'm certain that one day we'll succeed.